Hello everyone, welcome to our house, Illusion Hashtag, and in this lecture, we'll try to cover the topic Polymerase Chain Reaction. In polymerase chain reactions, it's simply what is this? In a simple word, if you want to copy your DNA, then the polymerase chain reaction is the only solution. It's one sub solution for you, the easiest one. And if you want to and there's no limitation. If you wanted to copy your DNA for a million times, it can do it. In short term, this reaction is called PCR, and in simpler, as we have said, this is the DNA photocopier. This is DNA photocopier. Means the technique which is making the photocopies of your DNA, and we have to make a science to make it available to the diagnostic industry. Thus, in simply, if we wanted to define PCR, that is the simplest technique which can make your DNA in a simply control C and control V in a more precise manner without any error. But error is always a part of anything. But the main agenda is to making it properly, precisely. And if you go with the recent past example, COVID-19 is a huge example where this technique used widely. In that time, we used RT-PCR. That is a variant of PCR. We'll go through this in a brief in my other lectures. But that is the thing. Apart from also for the diagnostic industry or in the field of also this forensic industry, this is a widely used method for identifying or just locating those people, those genomes. If you want to go with any method, any technique, any reaction, you need something. You need an environment, that is, you need an attitude to maintain. You need an instrument which will maintain everything, and some special ingredients means that is required for carrying out the reactions. And what are those? In simply, if you wanted to go for copy and pasting a DNA, that's what are the requirements for this? First things you need a template DNA. What is template DNA? Template DNA is nothing but the DNA strand you wanted to copy, the OG strand that you wanted to copy. That is a okay. This is a thing I need a multiple copies of these. Your parent copy. That is a template DNA. Now primers. Now primers is a very interesting thing. That is the most critical part, and this concentration and everything. This is very important. Now if you look at primers for copying this template DNA, somehow it need to be read properly. Now primers are the things. Suppose if this is your template DNA, a primer is a thing which can initiate your reaction. For initiating, as you all know, for initiating a replication, we need a 3' hydroxyl group. We all know. 3' hydroxyl group, this is required for starting one replication reactions. This primer plays an important role to binding to the side one the terminal portion of the template DNA, thus it can provide a 3' hydroxyl group and it can initiate the reactions. Next, DNA polymerase. Basically, this is a huge tool that is stacked DNA polymerase. Stack means thermally stable. And from where you can get this polymerase? You can get Thermus aquaticus. What is Thermus aquaticus? Thermus aquaticus is a algae which is basically found in the deep sea. From there, it is used to isolate these Thermus aquaticus. This is a tagged DNA polymerase. Normally, in our system, in our body, whatever DNA we have, that DNA usually is stable at 37 to 40 degrees Celsius. Uh, Beyond that, it will be dead. But if this Thermus aquaticus, these DNA polymerase, these enzyme is stable at around 100 to 110 degrees Celsius. They also show some stability. They will not degrade. They will not denaturation will not see in these enzymes. This Thermus aquaticus, this tag DNA polymerase, is one of the most critical and also the costliest thing for doing a reaction. Next, DNTPs. Why? What does it mean? DNTPs? Why? I'm not. Why this is called D? D for deoxy. Deoxynucleotide triphosphates. And we all know why deoxy. This is the base of DNA. If it's a riboxy, is oxy, it's not deoxy, then that is the base of RNA. It's more frequency 2-deoxy, more precisely we can say 2-deoxy. Thus, deoxyribonucleic triphosphates, those are things required for, those are the building blocks for any DNA strand. Thus, first thing is what? Template DNA. That is a thing they will follow. Okay, these are things I need to copy. Then the primers. Okay, primer isolate on the one thing. Okay, I am the thing. I bound to one terminal position. This is the 3 hydroxyl group. Thus, you can start the reaction. Now, we'll start the reaction. That is DNA polymerase. More specifically, tag DNA polymerase. That is thermally stable. Now, for doing the reaction, they need some building blocks. Who is the building blocks? That is DNTPs, deoxynucleotide triphosphates. Next, what we need? Buffer solutions. What is the requirement for buffer solutions? We all know for doing any reactions in a very optimized manner, we need to maintain the environments. Means we need to maintain a pH environment there. That is one of the most critical physiological factor for any reactions. This pH and we all know for replication also we know many metal ions such as magnesium we need. Mg plus one of the most major things we need magnesium. There's all those pH part, pH and ions, those are kept in the buffer solutions. This is important. And thermocycler. What is thermocycler? This is an instrument. This is an instrument will maintain all those environmental parameters. Means now we need 30 degree, now we need 40 degree, now we need 80 degree. Then again back to the temperature is not like that. Temperature is very simple. Now I need 90, now I need 50, 60, now I need 70. Again I need a pause. Then I need to go for this all those temperature thing. That environment it is maintained by thermocycle. Does you need to understand? Template is what? The thing it will copy. Primer will identify from what it to start. Polymerase will carry out the reactions. DNTPs, which are building blocks, will give the things okay, I am a conduit. And buffer solution maintain the environment. Thermocycler maintain the temperature. Thus, it will happen. Now, as I say, you are copying those DNAs. Thus, for making those things, there are multiple steps should be there. It's not like now I am one, now I am 10,000. It should not be like this. There should have some critical steps. Okay, 1 to 2, 2 to 4. It does understand it is this exponential reaction is not a normal linear. It's 2 to the order. That is 1 to 2, 2 to 4, 4 to 8, 8 to 16. It should be like this. And for maintaining these things, this is one cycle, two cycle. Each cycle there should have some specific steps for each cycle. What are those steps? First step, break it up. If you want to make some new things, you need to break it your previous toxic things. Next, you need to cool yourself down. Okay, fine, I break it up. I need to calm down myself. Thus, I can do a new thing. Then copy. Okay, that's one. Go for the new bonds. That is copy that and run it back. That is a loop. Let's go with the break it up. Means denaturation. It's simply like you are hitting those DNAs. Suppose here DNA strand, you are hitting it. Thus, it can split from a toxic relationship. And what is that? That is hydrogen bond. If you hit it, this hydrogen bond will break and it will turn into single strand. And this step is called denaturation. It's usually happen at 95 degrees Celsius. Clear? And after these things, now what is happening? In this DNA helix, you all know that is duplex. You are hitting it, and now it is going to single strand. Thus, in the single strand, the rest of the things can happen. And it can, that is a thing. I am a toxic thing, I am breaking myself, thus I make myself ready to be mingled. I am not mingled yet, I am single only. Next, cool vibes. That's it, you need to commit. Okay, I break it up. I'm right now I'm just trading myself out from this toxic part. I need to be ready to match myself with a perfect match. It's not like I'm matching with anyone. Randomly, no, not like this. We have to be very precise. We have to be very selective. While go for our next match. Okay? And this is called annealing. Means In this case, initially, suppose what is happening there, just sorry for this one. This is your DNA. You are hitting it up, thus it is became single strand. Means you previously or is a toxic relationship, this one toxic one. Now you are ready to mingle. Thus, in this state, this is happening at 95. Then you are hitting it and make it cool. Thus, oh, calm down, be polite, don't be anxious. Thus, you can be mingle with someone. That is then primer come to action and primer mingle with you in one terminal. Okay, I'm here. Just start your next reaction. And these things need to be very accurate, very precise. If this is precise, then your DNA, all the rest of the things will be precise. That's the thing. Okay, and this should be like I'm both sides, like you're matching your dating sites, dating apps. It's similar to this only. Both sides need to be right side only. One right, one left, it will not work. Both right, okay, we are perfect match. Go to let's go for the reaction. Next, copy. Means copy paste means here the tag DNA polymerase come into action. Tag. Okay, you got your reactions. Okay, you got your perfect match here. Now tag DNA polymerase come into action. Now it will go to replicate the things based on the three. Now, sorry for this disruption. This tag. Now tag, what will you do? Okay, you got your primer. And this is 3 dash and this is hydroxyl group. Okay, you got it now. Now tag DNA polymerase will come into action. And it will start extending the reaction, and based on this it will be parent strand and it will make the daughter strand. Thus a copy of this gene you will get. It's a complementary copy, but that is also okay, fine. And this is happening at 72 degrees Celsius. Here you can understand for the usual physiological polymers which we have in our system, they usually work optimally at 37 to 40, but titanium polymers work at a temperature of 72 degrees Celsius. This is the optimum temperature, 72 degrees Celsius. And annealing survive at 60 degrees, at around 57 to 60 degrees Celsius. Now what is happening here? It's simply, it was a toxic one, it break it up, okay, ready to mingle, it's a perfect match. Now building a relationship, there needs a building block, that is the DNTPs, and as you all know, for every relationship, the best friend plays a key role. The best friend is tachyon polymers. Tachyon polymers come and continuous reaction. Thus, the building blocks will slowly add and add, and it's a okay, you are not a very strong relationship. And tachyon polymers release. Means one piece, a perfect bond come, then best friend make a exit. And then run it back. Okay, bro, again, this is a toxic one. Run it back. Again, best friend commutation, everything will happen. And just repeat like for the TikTok trends, you all know. If some trends we like, we used to in Instagram while scrolling the things, okay, these reels I like very much. They used to see repeated, okay, how the dance move or how the song is moving, how the guitarist is playing. We just like to listen or sing for repeated times. And it is similar like this only. I will decide for how many loops I wanted to run, how many copy number I wanted to see. Usually we run for 30 to 40 cycles. That is the usual thing. And this same reaction, whatever happened like this, means break it up, annealing, and extension. It will again look for usually 30 to 40 cycles. After that, you will get some result from the graph and those this through some graph plotting that we'll understand on our next classes. Now, as you all know, we already said that in COVID we are using RT-PCR. Means PCR is okay, fine. That is a PCR. That is a broadly you can say PCR, but there is a various type within it. Thus, we can classify it in many types. What are the types? These are the broadly PCRs of 10 types. Among conventional PCR that we have already discussed, that is the thing. This RT-PCR, real-time PCR. Then understand, real-time PCR is shortly called qPCR. This is also reverse transcription PCR. That is also RT-PCR. The name are same almost, but the role is extremely different. In reverse transcription PCR, you will understand that R and T will be capitalized. But real-time PCR, both are never it will be capitalized. It will be small or it will be called qPCR. That is quantitative PCR. These two are widely used in the lab or in diagnosis work. These are the two things usually used or very much. This nested PCR, there's a double checking things, and in genomics, this is what I use. And these are other things. And I will go through all those PCR techniques in our different lectures. But usually, the widely used one and important for those master's level things, that is real time PCR, reverse transcriptase PCR, nested PCR, inverse PCR. We'll go one by one for the every topics. Not today, but in our next lectures. And that's all for today. Thus, today what we have learned? What is PCR? What are the things required for PCR? What are the basic steps involving this technique? And what are the key types, key variants we have in this market, in this scientific field till time? If you have any question, you just simply just ping me in my comment box. We'll surely will go there and just try to figure it out and try to solve it. And that's all. Try to share with your friends and every colleagues. Just we can make some more fun on PCR on different techniques. And that's all. Thank you everyone for your time.